Click for IB Biology. Here we're looking at absorption spectra, action spectra, and inductive reasoning in developing our understanding of the process of photosynthesis. There is a PDF in the description for you to annotate with your own notes and a quiz for you to use to check your understanding of the content presented here. We are going to look at two forms of experimental data. The upper section here is the absorption spectra, and this one here, that's the action spectrum. Let's discuss their difference. With the absorption spectrum, you can see that the x-axis is wavelength and the y-axis is absorption. Down here, we have the experimental setup. We have a lamp which is capable of variable wavelength. We can select the wavelength that we wish to transmit, A, and this will be passed through a sample of chlorophyll, shown here, and the light which is able to pass through the chlorophyll will be detected by this surface here, and we would call that transmission. So the absorption here, which is our y-axis, is calculated by taking the value of 100% transmission and subtracting the percentage transmission, which is here. When this is performed, what we discover is that the various pigments within our photosystems absorb light very highly in the region of the blue part of the spectrum and in the red part of the spectrum, but not in the green-yellow section of the spectrum. The second set of experimental evidence is what's called an action spectrum. In this particular case, the idea would be that we're going to change the wavelength and measure the rate of photosynthesis. So typically this could be done by having a plant, and we're going to shine light on this of the different wavelengths, first the blues, then the greens, the oranges, and then through to the reds, and we're going to somehow measure the rate of photosynthesis. So a classic way of doing this would be for an aquatic species. We could measure the evolution of oxygen being given off. We could collect that, the volume of oxygen over time, and calculate a rate in that way. There are various ways in which the dependent variable can be measured. When this is done, it's found that in the blue part of the spectrum and in the red part of the spectrum, we have high rates of photosynthesis. So notice the coincidence of patterns from the absorption spectra and the action spectra, which suggest a correlation between the absorption of blue light and the high rate of reaction with blue light, and similarly the low levels of photosynthesis with the green part of the spectrum, and once more higher levels with the red part of the spectrum. This is two sets of experimental data leading us to a hypothesis about how these chlorophyll molecules are operating, and we see a correlation. Going from experimental work to a hypothesis is known as inductive reasoning, inductive logic. Although we have a suggested correlation, we would need more experimental evidence to be able to claim a causal relationship between the two. That is to say that these pigments here are causing this high rate of photosynthesis. We don't have the experimental evidence for that yet. We'll look for that in our theory, which will follow this video. The next video in the series, as we go into the details of the biochemical pathways, are now appearing in the links below, along with the subscription button and a link to the playlist, so that you can look at the rest of the videos in this particular sequence on photosynthesis.